How many of you have a friend or a family member who has diabetes? Most of us, right? I work on diabetes, and I also come from a family of diabetics. My lab works on diabetes-induced heart failure. The adults with diabetes are more likely to die of heart failure than other adults. And our research is preclinical, and we use type 2 diabetic animal models. Here you see a cell from the heart of the rat. So the cells have lots of proteins on their surface, and they perform various functions. One of the protein on the cell surface is a transporter, a glucose transporter. It transports glucose from blood into the cell. But this transporter works with insulin. In diabetes, these transporters go less with inefficient insulin. Some of the diabetic treatments actually attempt to increase these transporters on the cell surface. Usually, our body adapts to changes. One such compensatory mechanism in diabetes is increasing another form of transporter. That one also brings glucose into the cell, but at the same time, it also brings in lots of sodium. So what's the result of sodium? Increase in sodium inside the cell in diabetes. Sodium, that is the first ion in our tail. Now, let's move on to mitochondria and see the effect of sodium inside the cell. So mitochondria are the powerhouse of the cell. For an organ like heart, which continuously pumps blood, it needs lots of energy. And mitochondria produces such energy, but while producing energy, it also creates some free radicals as a byproduct. Luckily, mitochondria have their own antioxidant systems, and they scavenge the free radicals. But for regenerating these antioxidants, mitochondria needs calcium. That is our second ion in the tail. So when mitochondrial calcium is there, then less free radicals and more antioxidants. But what happens with increase in sodium is one of the protein on the mitochondria gets active, and it takes mitochondrial calcium away from it. So when the calcium inside the mitochondria goes less, more free radicals, which is bad for the heart, and free radicals can trigger some mechanisms that lead to heart failure. What we found in our research is by blocking the exchanger protein, we could actually increase the mitochondrial calcium, reduce the free radicals, and suppress the mechanisms that trigger heart failure. This is clinically significant because this can actually mean reduced incidence of heart failure in diabetic patients. So our research is preclinical, but if this is proven clinically, then this can actually translate into mitochondrial calcium targeted therapeutic strategies for addressing heart failure in diabetes. Thank you.